Have you ever wondered how a wound heals under a band-aid? What is happening inside your body that causes so much change from your childhood to adulthood? How do you look so similar to your parents? Well, the answer to all those questions above is mitosis. So, what is mitosis? Mitosis is a crucial process in the growth and repair of multicellular organisms as it results in the production of identical daughter cells. This process ensures that all cells contain the same genetic information as the parental cell. Mitosis is a series of orderly events that leads to the equal distribution of genetic material to the new cells. Traditionally, mitosis is divided into 4 phases: prophase, metaphase, anaphase, and telophase. It is important to remember that mitosis is just one part of the cellular cycle, during which cells go through metabolic activity, DNA replication, chromosome segregation, and cell division. To start, let's focus on the G2 phase of the interphase, where several key events take place to ensure the cell is ready for division. At this stage, a crucial feature is the presence of a nuclear envelope that encloses the nucleus. Simultaneously, the cell undergoes the duplication of centrosomes. Centrosomes are pivotal regions in animal cells responsible for organizing the microtubules of the spindle, a structure critical for proper chromosome segregation during mitosis. Of course, please note that during the G2 phase, chromosomes that were duplicated in the preceding S phase cannot be observed individually. This is because they have yet to condense, highlighting a critical step in the cell division process. As we transition into the dynamic process of mitosis, our first stop is prophase. This initial stage is marked by a series of intricate events that set the stage for the precise division of the genetic material. During prophase, the once diffuse chromatin fibers undergo a remarkable transformation. They become tightly coiled, condensing into discrete chromosomes. These chromosomes become observable under a light microscope. As a chromatin condenses, each duplicated chromosome takes center stage. Here, it appears as 2 identical sister chromatids joined at their centromeres. This connection is often extended along their arms by cohesins, resulting in what we term as sister chromatid cohesion. A critical player in prophase is the mitotic spindle. Named for its distinct shape, the mitotic spindle is composed of centrosomes and the microtubule extending from them. As the mitotic spindle forms, the centrosomes, which were duplicated during the interphase, start to move away from each other. This movement is facilitated partly by the lengthening microtubules extending between them. As our journey through mitosis continues, we arrive at the metaphase. In metaphase, the cell undergoes critical changes that bring us closer to the ultimate goal of precise genetic division. At this stage, the centrosomes, which have been on a journey moving away from each other during prophase, are now positioned at the opposite poles of the cell. This spatial arrangement is vital for the subsequent events that unfolds in the mitotic processes. A defining feature of metaphase is the alignment of chromosomes at the metaphase plate. This imaginary plane is equidistant between the 2 poles of the mitotic spindle. The chromosome's centromeres specifically lie at this metaphase plate, creating a symmetrical arrangement. The precision of chromosome alignment is achieved through the attachment of kinetochores to microtubules. For each chromosome, the kinetochores of the sister chromatids are attached to kinetical microtubules coming from opposite poles of the spindle. This intricate attachment ensures that each chromosome is poised for equal segregation during the subsequent stages of mitosis. This process sets the stage for the impending separation of genetic material. As we progress further, we arrive at anaphase, the brief yet decisive stage that marks the separation of genetic material. Anaphase kicks off with the cleavage of cohesin proteins. This cleavage liberates the 2 sister chromatids of each chromosome pair, allowing them to part suddenly. This transformative moment marks the transition of each chromatid into an independent chromosome. As the cohesin proteins are cleaved, the 2 new daughter chromosomes, now free from their sister chromatids, embark on a journey toward opposite ends of the cell. This movement is facilitated by the shortening of kinetical microtubules attached to the centromere region of each microsome. By the clivination of anaphase, the two ends of the cell now harbor identical and complete collections of chromosomes. This symmetry is a testament to the precise and orchestration of the mitotic process, ensuring the faithful distribution of genetic material. Now, as the cell enters telophase, the final step of mitosis, key events include the formation of two daughter nuclei, facilitated by the emergence of nuclear envelopes. Nuclei reappear, indicating the restoration of functions related to protein synthesis. Chromosomes decondense, and any remaining spinal microtubules are depolymerized, concluding their role in guiding chromosome movement. This resolution and renewal phase marks the completion of mitosis. The cell is now prepared for its next cycle of growth and division. Now, as we transition from theory to hands-on application, if we would like to observe different phases of mitosis, how can we let that happen? With our naked eyes? Of course not. A possible approach, however, is to use the microscope to observe the cells of garlic root tips. More precisely, there are 4 stages: dissociation, rings, staining, and slide preparation. In the dissociation stage, we begin by cutting 1 cm of active garlic root tips. These pieces are then immersed in a 3:1 ethanol to acetone fixative solution for 3 minutes. The result? At this time, the cell was killed by dissociation, so the cell stays in a different period of division and will not change and maintain the original shape. Cellular processes come to a halt, preserving components in a lifelike state. The purpose here is to separate tissue cells from each other in a soft way. Moving on to the ring stage, the fixed roots are washed in distilled water for several minutes. The purpose is to wash away hydrochloric acid to prevent excessive dissociation and to facilitate staining. Well, if the dissociation time is too long, the chromosomes may be destroyed, and vice versa, if the dissociation time is too short, then the tissue cells are maybe not sufficiently dissociated and cannot be separated from each other. In the following staining stage, roots are stained with ginseng violet for 3 minutes, followed by washing off excessive stains using water. Ginseng violet's purpose in this context is clear. It selectively binds and colors chromosome structures, making them visible for observation. Then, in the slide preparation stage, we place a single root on a microscope slide with water. By applying a cover slip and gently tapping the slides, the root tissues is squashed into a thin layer, so that the observation of the mitotic chromosome will be much more recognizable. Now, by placing the prepared slide containing the squashed garlic root tissues onto this microscope stage, we are free to observe based on our preliminaries of mitosis. What may be obtained? Well, chromosomes in various phases of mitosis will become visible under the microscope.